Hello, everyone, and welcome to the FY22 TRAP Kickoff Meeting. My name is Chad Jackson. I'm the Senior Director of the Preclinical Translational Research Program at the Foundation Fighting Blindness. I want to welcome you here today, and I hope that you enjoy this session as much as I will. Before we get started, I would like to briefly review a few details for the call. Currently, all participant lines are on listen mode only, with no video. Today's conference is being recorded and is available with closed captioning. To activate closed captioning, please select the live transcript option located at the bottom of the Zoom interface. Then select Show Subtitles. If you are using a screen reader, please be aware that the controls are at the bottom of the Zoom interface. The control bar may collapse when it's not in use. If you prefer to prevent the controls from auto-hiding, go to Settings within the Zoom platform, select Accessibility, and then select Always Show Meeting Controls. It might be helpful to maximize your window and navigate by using the Tab key. Additional buttons and settings are available by pressing the Alt key. During the call, you may ask questions through the Q&A chat or by sending in an email to info@blindness.org. Again, that is info@blindness.org. We will address questions during the Q&A sessions following each presentation as time allows. Any questions not answered here or submitted to info@blindness.org will be answered by email following the webinar. The Translational Research Acceleration Program, known as TRAP for short, funds the most promising preclinical research, helping to move technology from the bench and toward clinical utility. The research we seek to fund in this program is prioritized in three research priority areas. One is novel medical therapies that covers small molecules and biologics, genetic technologies that cover viral and non-viral therapies, which includes gene editing, and regenerative medicine. These projects are placed into an aggressive program management plan to accelerate research toward clinical trials by continuously monitoring the research progress and streamlining all areas to either meet or exceed the research and development timelines. The research plans are anchored in milestone accomplishments, fostered through a close relationship between the project and an industry experience mentoring consulting. Our goal is to establish enough data to foster the relationship with new partners and venture capitalists to help move the technology toward regulatory filing and hopefully in the establishment of a clinical trial. For well over a decade, the Foundation Fighting Blindness has made translational research a line item funding priority for the organization. Over that period, the foundation has funded numerous research projects that has ultimately led to many clinical trials and potential therapeutic assets, as well as companies that are all focused on addressing inherited retinal disease and dry age-related macular degeneration. For example, TRAP awards made to researchers such as Drs. David Gamm, John Flannery, and Shannon Boyd aided in the establishment of Opus Therapeutics, Videri Bio, and Atsena Therapeutics, respectively. This motivates us to continue to advance the research for inherited retinal disease and dry age-related macular degeneration. None of this would be possible without the generous donations and continuous engagement with the Foundation Fighting Blindness by our constituents and stakeholders. Therefore, today we bring you four projects that have started this year to help shepherd potential therapies toward clinical utility. And here are some of our FY22 award winners. Today, you will hear from Dr. Marius Adder, Dr. James Hurley, Dr. Roy Chen, and Dr. William Beltran. Unfortunately, as previously listed, Graybug Vision will not be able to join us at this meeting today. I would like to personally thank everyone for taking the time out of the day to join us on this call, and I hope that you all learn and are invigorated by what we hear from each presentation. Again, there will be a Q&A session after each presentation. Therefore, please put your questions into the Q&A box. At the end of the sessions and the question part, Dr. Claire Gelfman, the Foundation Fighting Blindness's Chief Scientific Officer, will give closing remarks. Now, our first presenter, is Dr. Marius Adder from the Technical University of Dresden. Dr. Adder is focused on retinal degeneration due to loss of photoreceptors and understanding the prime causes that drive these conditions. His experimental work focuses on development of cell-based therapies to treat lost cells in the retina, including photoreceptor cells and retinal pigment epithelium. The title of his TRAP project is Reconstruction of the RPE Photoreceptor Interface via Sequential Transplantation of HLA homozygous iPSC, RPE, and photoreceptors. Dr. Adder, the floor is yours.
Thank you very much. I will open my presentation. So, do you see my first slide?
Yes.
Yeah.
Good. Yeah, thanks very much, Chad, for the kind introduction and for the invitation for this exciting conference to show our TRAP project. As you said, my name is Marius Adder. I'm from Dresden, from the Center for Regenerative Therapies in Dresden, Germany. And we directly go into the topic. So as most of the listeners and viewers will know, the retina is where light is detected and where vision starts. And actually they start with the photoreceptors, the light-sensitive photoreceptors, rod and cones. And if these are lost, that leads to retinal degeneration. However, the support cells, the retinal pigment epithelium, is also absolutely essential for proper function, and also dysfunction or loss of RPE cell leads to retinal degeneration. We see that in diverse retinal degenerative diseases like retinitis pigmentosa or also AMD. Our approach is now, or our question is, can we maybe by injection or transplantation of proper cell populations reconstruct or rebuild photoreceptors or even RPE cells or both together? So what we have in the last years have established is a pipeline for human photoreceptor transplantation. So what we are doing is we use human-induced pluripotent stem cells and generate so-called retinal organoids out of these. And then we use a reporter in the photoreceptors to sort out photoreceptors, cones, or rods, or both, to have a suspension with photoreceptors that we are transplanting in the so-called subretinal space in mice with retinal degeneration. One example I want to show you today is an example of a cone transplantation. For this, we use a mouse model that is called CPFL1, that stands for cone photoreceptor function loss. And that means that in these retinas, the structure more or less is still okay, but in these mice, we don't have any cone function or cone photoreceptors. So they are partially blind. And if you now take these photoreceptors at a specific time point, actually from organoids that are 200 days old, and transplant them, you see an amazing integration of the human photoreceptors here in green, integrated into the outer nuclear layer of the host. And you see even more that there are here in pink protrusions developed that remind us about so-called inner segments. And indeed, if you look now in a higher magnification using electron microscopy, you see on the one side the host rod photoreceptors, and directly beside the human photoreceptor transplant, the cone photoreceptors. And here you see the inner segments filled with so-called mitochondria. And even more, if you wait up to 26 weeks, you can see even a very important compartment of the photoreceptors that is called the outer segment. Actually, outer segment is where the light sensing process really takes place. And we could find these transplanted human photoreceptors generate outer segments. To be functional for photoreceptors, the signal that is generated in the outer segment has to be transmitted via a synapse to a secondary neuron. And indeed, we could also find synapses in the transplants. And they have the special structure of photoreceptor synapses that are called the ribbon synapses. So they have a ribbon and vesicles around here. So beside these structural maturation that we could see, we also saw that the donor cells were really integrated in the tissue as they were enwrapped by endogenous Müller-Glial processes. And this is very important for any function, but also for the maturation of these cells. With this, we asked the question, can these photoreceptors even be functional? And we teamed up with Günther Zeck and Miriam Reh. And they are experts for a measurement that is called microelectrode electrode array, MEA. And that means you can put out a retina flat mount, put it on these microelectrode arrays, and that are hundreds of little single electrodes that allows you to measure functionality of neurons in parallel and several hundred simultaneously. So that is a scheme that you are measuring not directly the functionality of a photoreceptor, but if the photoreceptor sends signal via the bipolar cells to ganglion cells. And that is how it looks like in a transplanted retina. This is taken out from the eye on these microelectrode arrays. Here you see the fluorescence transplanted cells. And the interesting outcome is that indeed only in areas here in yellow where a transplant was, there was much more activity of on and off retinal ganglion cells. And importantly, if you knock down or suppress, inhibit all functionality of the rod photoreceptor pathway, only in transplants you still have activity in the off pathway, showing that the transplanted cone photoreceptors really perceived light and could send the signals to the ganglion cells.
Dr. Adder, I'm sorry, let me interrupt just for 1 second. There may be attendees on the line who are visually impaired. So if you could please describe the figures, and you're talking about either the increases or decreases in those figures, could you please verbally describe those during your presentation? Thank you.
Okay, sure. Thanks for mentioning this. So to sum up, we generated a pipeline using human retinal organoids, transplant the photoreceptors in partially blind mice, and these transplanted donor cells showed extensive incorporation into the mouse retina in the ONL. They showed a clear graft polarization with inner and outer segments. They had a close interaction with the host Müller-Glial cells. They showed contact to bipolar neurites, and they showed graft-mediated light responses. So sum up, we can indeed now or have now technology to replace photoreceptors in the degenerative retina. So what is with RPE cells? So there's already workouts already in clinical trials even where pluripotent stem cell-derived RPE has been transplanted. So, and we also wanted to go in this direction, and what we used is a mouse model where we deplete completely the endogenous RPE cells by systemic injection of sodium iodate that leads to complete loss of endogenous RPE cells, so the mouse cells. And 1 week later, we used then again iPS-derived RPE cells for a transplantation to see if they can replace the RPE monolayer. And indeed, what we compared were 2 different populations of RPE cells that you can't really distinguish within the culture. So it's the same culture, just passage 1 time. And in the passage 1 transplants, you nicely see here in red the human transplanted cells, the RPE cells, and you see large areas that look like a monolayer. So they really formed a monolayer that is in contrast to these smaller areas where you have more cluster of cells. And that is in striking difference to the second population, also RPE cells, also from the same culture, but these cells massively formed clusters. They survived as good, but they formed clusters and not these monolayers. And if you now look at the retina, how it looks like, the retina looks very bad beneath the clustered RPE cells, in contrast to the monolayers where the outer nuclear layer or the host retina looks pretty good. So there is It's not about only survival of transplanted RPE cells, you need the right subpopulation. So with other words, we could form RPE monolayers by a RPE subpopulation by transplantation in this model. Our question is now, if we can transplant or replace RPE cells and photoreceptor cells, can we replace both? And that is actually the TRAP project that we have applied for. And how we call that is a sequential co-transplantation of RPE and photoreceptors. So what we are using is a genetic photoreceptor degeneration mouse model that additionally will be depleted by our injection of sodium iodate, so that you have a depleted retina without photoreceptors and without RPE. And then first we will reconstruct by transplantation of RPE suspensions, the RPE monolayer. And then later, 3 weeks later, we'll additionally transplant photoreceptors, and then we'll analyze the eyes. And actually we have a proof of concept for this sequential transplantation system. Here you see labeled in blue the human photoreceptors and the human RPE that made a monolayer, and beneath the photoreceptors. And also in these photoreceptors, we could see stainings for inner segments with the right polarization that was confirmed already in preliminary data, I have to say, at the electron microscopy level with a human RPE monolayer and human photoreceptors and inner segment formation. So what we now want to do in the next 3 years with this grant is we have to look for several aspects on the road towards the clinical application of such an dual transplantation approach. First, we have to in detail analyze the RPE photoreceptor transplants in terms of differentiation, maturation, and functionality. And that means at the end of the day, we have to optimize this RPE photoreceptor co-transplantation system. Another very important aspect is how can we purify our transplantable RPE cells and photoreceptors. As I told you in our work up to date, we used a reporter to sort them, or just took the RPE cells out of the culture, but that would be not applicable for clinical use. An alternative for that would be to use so-called cell surface markers to enrich and purify for the target cell population. And in the last years, we had a grant together with a company that is called Rna Biotech, and they are actually real experts on cell surface markers and sorting. And they looked for us and looked for cell surface markers that are specific for subpopulations of RPE cells, but also for photoreceptors, rods, and cones. And for the RPE cells, we have now first results. So they identified target cell surface markers for a target cell population within the RPE cells. And here you see flat mounts of retinas after 3 weeks after transplantation. And you see loads of surviving cells in the unsorted, the target, and the non-target fraction. However, a really good and fine monolayer you mainly see only in the target fraction. So we have now already for the RPE cell surface markers detected that are very good for making RPE monolayers after transplantation. Another very important aspect is the immune reaction or response of the eye. As you know, for the transplantations, you have to find fitting organs. If you talk about organ transplantation, also in cell transplantation, you have to think about it. Though the eye is immune privileged for some, in some way, it's not completely immune privileged. So we need to find cells that can survive in many patients. And here we are following a route where we use so-called super donor stem cells, pluripotent stem cells. That means one stem cell line can be used for many, many different patients. So they are so-called HLA homozygous IPS line, and we will now try within this grant to generate RPE out of these lines and also photoreceptors, and then use them for transplantation. So for taking together what we want to do in the next 3 years, we want to make a preclinical establishment of sequential co-transplantation of cell surface marker purified human RPE and photoreceptors generated from HLA homozygous IPS cells. With this, I'd like to thank you very much for your attention, and I'd like to thank my group and our sponsors, and I'm happy to take questions. Thanks.
Dr. Adder, thank you so much for that. We are actually running a little bit late, so what we'll do is we'll encourage people, please put your questions into the Q&A box. There will be a general Q&A session at the end of all the presentations, so Dr. Adder, we will then circle back for the questions that are directed towards you. Thank you for that presentation.
Thank you.
All right, our next presenter is Dr. Roy Chen from Baylor College of Medicine. Currently, Dr. Chen is a professor at Baylor College of Medicine in the Department of Molecular and Human Genetics. He has been with Baylor College of Medicine's Human Genome Sequencing Center for almost 20 years and has been directly involved in many genome sequencing and technology development projects. In the last 5 years, his group has cloned 7 novel human LCA and RP genes and performed extensive functional studies of the disease mechanisms. Their retinal disease next generation sequencing capture panel has been used for molecular diagnosis for more than 7,000 patients with various retinal diseases worldwide. As a result, they have established one of the biggest retinal patient collections that aids clinical trials. The title of his presentation is Retinal Regeneration through Epigenetic Therapy. Dr. Chen, we are ready for your presentation.
Can you see my slide?
Yes, we can.
Okay, thanks. First, I'd like to thank so my name is Roy Chen, and then from Baylor College of Medicine, as introduced by Chad. I would like to first thank the Foundation Blindness for its support, and for everybody here, and for your generous support and then effort in this field. So I would like to share you today about some new project actually we started in the lab. So to this audience that it's not important, to this audience, I don't need to stress on the importance of the vision impairment in terms of human disease. This is one of the top 10 disabilities in US. I think what's make matter worse is it's expected that the vision impaired will grow dramatically in the next 20 years as the population age. So currently, there's many treatment that has been developed to treat the vision impaired diseases, but most of them have some limitation. Just a quick, list of the major age-related retinal degeneration disease, such as diabetes retinopathy, age-related macular degeneration, and glaucoma. And then currently there are treatments, for example, like anti-VEGF and laser treatment. The problem with those treatment is they really not targeting the real cause of the disease. And then generally they can slow down the progression of the disease, but eventually a lot of patients develop resistance to it, and then eventually they still disease will progress to a point that they will have lost a lot of vision. So another big problem is if you cut this late, and then when the neurons are already degenerated, then there's those treatment. None of those treatment will be useful or effective. As the previous speaker, Mira showed you very nicely that alternative way to deal with this is looking for stem cell related method therapy. Currently, there's a lot of this clinical trial already going on using RPE. Stem cell to RPE treatment, and then with some very promising results. However, this remains very challenging to using this same way to treating neurodegeneration, as alluded by Maris. Although it has been, there's a lot of progress, but most of the transplanted cells die short after, particularly in the primate situation. And also the surviving transplant cells really need to overcome the challenge to differentiate and integrate. And thirdly, of course, the cell transplantation have an immune response that you need to have immunosuppressant for a long time. So to address that challenge, regeneration seems an ideal alternative therapy for this sort of late stage disease. So there's several key advantages. The first one, the source of the neuron cells, is endogenous to the patient. So therefore, there's far less likely to be rejected or induce any immune response, rejection. The second, the newly generated endogenous neurons can be efficiently integrated into the functional circuit and lead to a full restoration of the function, as already shown happen in the nature. I will show you in the next few slides. The third one, there's no need to perform invasive surgery for cell transplant, which really reduce the risk of damage and the cost of the treatment. So regeneration is a gift from the nature. We already, as I showed in this slide, the retinal regeneration can be robustly induced in zebrafish. So the left panel are sort of control no injury the retina of the zebrafish. To the right of the panel are injured, and then this cross day after injury. So when you have an injection, say injury induced by laser injection, laser shining, for example, as you see in the injured part, you see degeneration of the photoreceptor cells and other cells in the retina. And then when you go over, like 6 weeks later, you can see actually go back to like wild type. So the cell in fact can respond to that damage and then to regenerate all the cells and back to just nothing happened, like nothing happened like wild type. So unfortunately, this sort of ability to regenerate is not exist in mammals during evolution. So people use the fish as a model to study where the cell come from, what's the source of the cell to regenerate, and then by tracing different cell types in the retina, and then they actually identify the mural glial cells in the retina is the source of regeneration. So as I mentioned, the regeneration of the retina is sort of absent or shut down. That process is disabled in mammals. So people have been thinking about now, how can we turn that system back? I mean, how can we use that the system and then turn it on in the mammals and then to as a therapeutic method? So the landmark paper in 2017 by Nature from Tom Ray's group, they were able to express two markers like ASC1, a early neural progenitor cell markers or genes and transcription factors, ASCL1, and coupled with HDAC inhibitor. And what they show that by doing this, you can actually basically move the mural cells in the retina upon damage, and then let them be differentiated, and then they can give rise to neuronal cells. So since that paper, several groups like Beltran's group and Tom Seth's group, Seth Brackshaw's group, have also published additional manipulation with different combination of transcription factors that can improve that efficiency. However, overall, the regeneration is still quite limited, and primarily in the inner nuclear layer of neurons. It's really hard to make RGCs or photoreceptor cells. And then also, one really drawback is those genes need to be expressed prior to the damage, so you can prime the mural glial cells, make them ready. So in this case, you can think this is really not going to be suitable for therapy per se. And the third one is the overexpression of, you really require overexpression of a lot of genes that normally not expressed at that time. The exogenous gene required that could have a unintended side effect. So we have been thinking about instead of use sort of overexpress individual transcription factors and the other markers, is that possible to really switch this regeneration state more easily, or think about maybe this modulator in the global transcriptional state that actually responsible to shut down this regeneration process in the mammals. So our hypothesis is like maybe we can induce robust and comprehensive retinal regeneration in mammals through global transcription regulation. So I've been looking at different molecules to modulate the global gene extraction program. So luckily, what we find is this one particular compound that generate the through collaboration here at Baylor. It's called SYC1127. That when we inject these particular small molecules, as you probably can appreciate, the upper panel is the wild type one, injected with PBS, and then the lower one is a mutant, specific mutant, which typically have degeneration of photoreceptor cells, is one of the LCA disease genes. And when we inject the compound, you can see a proliferation of all three layers in the mouse. So when you inject the same compound in the wild type mouse, there's no effect, nothing happened. So when we do a close look at what's those proliferated cells, what they are, as you can see, the left one is the PPS injected. It's just like wild type, and then these particular things we look at RPPMS, which is standard RTC cells, which previously really hard to regenerate, and then use other method. As you can see, there's a thickening of this region, and then if you stand RPPMS, you clearly see multi-layer of RTC cells basically are induced. So so it's really also the Mueller cell we are trying, we also induce the regeneration. The answer is probably yes. So when we look at the SOX9 markers, which basically highlight all the Mueller glial cells, in the wild type, they form a very nice line in the middle of the inner nuclear layer, and in the injected mouse, you can see the number of the SOX9 positive essentially increased about 50%, and then you can also see the Mueller glial cells get more activated by GFAP staining. And not only the number go up, they also consistent with that, and also we see the EDU staining, which basically tells you cells are proliferation. So and then also we see cells which are proliferation and with Mueller markers and also RTC, suggesting those cells are transitioning into the RTC. So therefore, in our system, it's also we use the Mueller cell as a source to regenerate neural cells. So based on those data in the TRAP project, so our aims and milestones are we would like to find out where it's really, further identify is really the Mueller cells is the only cell resource for the regeneration, and then what question is basically examine the regeneration potential in different degeneration model. So the model we have using is the genetic model. So what happened to the damage, to the mechanical damage or chemical damage. And when we want to find out the treatment window, when the regeneration can be induced robustly at different age, use the mouse as model. So by answering these three questions, our hope is to really establish, to finish the preclinical study, establish epigenetic manipulation as a very suitable approach to induce retinal regeneration therapy. So in summary, we have achieved really robust retinal regeneration in mammals. And the second one is the retinal regeneration is induced by modulation epigenetic modifications, which is a very transient application of small molecules. So in the mouse we showed you, it's really only treat once or twice. So it's really just two quick injection. And third one is our finding is highly potential to lead the development of novel therapy with broad impact because you really can regenerate all cell types in our hand at this point. So knowledge for people who have done the work. It really was initiated by a Richard with a former PhD student, and now it's carried out by Selma and Jiaxiong with current postdoc in the lab. And then this is in collaboration with Fang Ru and Yangcheng Su, who basically doing the compound optimization and the synthesis. And then we are funded by Foundation by Blindness now, and also by several other funding agencies. Thank you very much for your attention.
Dr. Chen, thank you so much for your presentation. Really appreciate it. We do have some questions ready for you, so we'll take a few minutes and address those. The first question is, if the tissue has a gene mutation or the cells, won't the regenerated cell type have the same mutation?
Yes, so that's a very good question. So I think there's twofold. One is, of course, the Mueller cell have the exact same mutation as the say photoreceptor cell that degenerated. And then I think there are twofold. One is you could do gene therapy to first change or fix the mutation in the Mueller cells before you induce regeneration. The second one is also many of degeneration diseases are actually very long time. It takes a very long time. So I will guessing if you have a fresh made, say, photoreceptor cells, it will last another 20, 30 years, which is also quite useful in that case.
Thank you. The next question is, with this type of therapeutic approach, would it be dependent on the stage of the retinal disease?
So we haven't explored all the kind of all degeneration models, but from our preliminary data is that it seems the therapy is useful for different model. And then we'll imagine even the photoreceptors are entirely gone or nearly entirely gone, this should be still useful. So this is designed for late stage, therapy.
Late stage therapy. All right. Thank you so much. The last question is just, I think, more for maybe educating our audience. And can you sort of explain what are mirror glial cells and why are they so important or special in this case? Why aren't other cell types were being targeted for this therapeutic approach?
Yeah, the mirror glial cells, part of the retina, its normal function is the several major function of the mirror cells. One is structural cell. So it have very long processes across the whole entire retina. The presence of mirror cells is important for the structure, integrity, or stability of the retina. The second one is also sort of glial cells is wrapped around, say, photoreceptor cells like cones. It's really important for the retinal cycle for the cones, for example. So the mirror glial cell was differentiated or produced by the progenitor cell. Is when you have, it's interesting, like the photoreceptor cells, all the photoreceptor cells or the other retinal neurons are derived from the same pool of progenitor cells, retinal progenitor cells. So the progenitor cell, when it writes all kind of neurons, when it goes to late stage, it become like, you can say the leftover, progenitor cells become mirror glial cells. So mirror glial cells have a lot of similarity in terms of transcriptome and the state as the progenitor cells. That's why we think why it's easier to reprogram it and go back to the more undifferentiated state and then give rise all the neuronal cells.
Thank you. And thank you so much for your presentation. We really appreciate it.
Thanks, everyone.
All right. Our next presenter is Dr. James Hurley from the University of Washington. The Hurley Lab focuses on understanding how energy metabolism is controlled in photoreceptors. This energy balance is key to understanding how the retina functions in different states. And Dr. Hurley received his PhD from the University of Illinois in physiology and biophysics. The title of his presentation is Stimulation of Fatty Acid Oxidation to Diminish Drusen. So this talk will be focusing on sort of ways to mitigate age-related dry macular degeneration. Dr. Hurley, the floor is yours.
So is that sharing now?
Yes, it is.
Yep. Okay, so my name is Jim Hurley, and as Chad mentioned, our lab is at the University of Washington in Seattle. And I'm also very, very grateful to the Foundation of Fighting Blindness for their support for this project. I'm going to tell you about our plans to stimulate fatty acid breakdown to diminish drusen. And as probably most of you know, in macular degeneration, people often get these drusen deposits. So one of the which are shown here in a fundus photograph of someone with a certain type of macular degeneration. I think you can see these drusen deposits here. And so one of the leading researchers in studying macular degeneration, Christine Curcio, often likes to refer to these as oil spills. And she calls them oil spills because they're essentially they're very enriched in fatty material, lipids. And so I think that's a very useful way to think about these, a useful perspective. And oops. Yeah, and so because there's oil spills can mean a couple of different things, and so one possibility is that an oil spill, oil can be spilled from your car, and you see an oil spill on the driveway as an indicator, as a symptom that something was wrong with your engine. But the other significance perspective on oil spills is that a tanker could break up and release oil, and the oil could cause damage to the environment. In this case, destroying a mangrove forest. And so, I'm a relative newcomer to this field, but it seems from what I've learned is that it's not really clear whether Drusen are a symptom of something going wrong, or whether they're a cause, or whether they're both. And so, one of the things that we're hoping from this project is we'll get some insights into that. So, where do these oil spills occur? So, as you've heard in earlier presentations, and probably most of you are aware, the retina in the eye is made up of it's comprised of various layers. And one of the really important layers is in the very back of the eye, where the blood flow is, the choroid, or this capillary bed that provides blood, and that's what provides the nutrients to the retina. It all comes from that blood for the outer retina, where the rods and cones are. And so the nutrients have to flow through a layer of collagen, which is referred to as Bruch's membrane, and then that has to flow through then they have to flow through a monolayer of cells that you heard about earlier called the retinal pigment epithelium. And then those nutrients can eventually reach the rods and cones. So those include things like glucose and oxygen. So this oil spill, when it accumulates, most of it accumulates in this region right here between these monolayer retinal pigment epithelium cells and this collagenous layer. There's other places where the oil accumulates also. But I think most of it starts accumulating in this region, and so you can imagine that could interfere with the flow of nutrients. So that's one idea about what it might be doing. So here's a higher resolution version of that diagram from a paper by Christine Curcio, who's thought about this a lot, thought about the role of lipids in Drusen and AMD quite a bit. And so what she shows in this diagram is that the drusen can accumulate in this region, like I said, between the pigment epithelium and this collagenous layer called Bruch's membrane. If you look at it a little bit closely, what people have realized, and what she's really highlighted in her research, is that a lot of the drusen material is made up of these structures that are called lipoprotein complexes. And you've probably heard a lot about lipoprotein complexes because they carry things like cholesterol around in your blood. And they transport the fats and nutrients from and cholesterol from your intestines to where the fat is stored. So these are the HDLs and LDLs that you've probably heard quite a bit about. But the important thing that I want to mention about these lipoproteins, these complexes that float around in your blood, in this case in drusen, is that they have a hydrophobic interior. Fat. The interior is loaded with fat, and then there's a shell of what are almost like detergent molecules, phospholipids that are around the outside. But if you look at what those are comprised of, what they're made of, so the interior has triacylglycerol, which has 3 fatty acids, and it also has a esterified cholesterol, which has 1 fatty acid. And the outer shell is made up of phospholipids that has 2 fatty acids. So the point I'm trying to make is that the fatty acids are a major component of these lipoprotein particles, and lipoprotein particles are a major component of the drusen. So there's another disease that's really been widely studied because it's a very prevalent disease. I think I memorized this. I think it's like 12% of the U.S. population has some form of non-alcoholic fatty liver disease. And if your diet contains too much fat, that predisposes you to get this disease. But it exists in various stages. But if you have a healthy liver, you have small droplets of fat in your hepatocyte, your liver cells. But if you're starting to have non-alcoholic fatty liver disease, these droplets of fat get larger, and eventually they can cause inflammation and lead to cirrhosis. So this is it's a little bit similar to what we were just talking about with this accumulation of drusen. And so turns out, since this is such a prevalent disease, there's several drug companies that are working on developing pharmaceutical agents to try to diminish the amount of fat, and then diminish the amount of inflammation in these liver diseases. And so 2 of them in our one called Fursocostat by Gilead, and then more recently we've gotten interested in a drug called Clisacostat made by Pfizer. And both of these drugs are being developed by these companies to treat non-alcoholic fatty liver disease or non-alcoholic steatohepatitis. And the approach that I'll show you what the activity of that enzyme is in a minute. And they're pretty far along. They've gone through phase 1 and phase 2 trials, and they're safe. They're not FDA approved yet, but they've been found to be safe in those trials and effective in humans in diminishing the amount of fat in the liver and diminishing the amount of inflammation. And like I said, neither is yet approved by the FDA, but actually just a couple of days ago, I was checking up on this, and it turns out that the FDA has approved it for their fast tracking protocol to try to get them approved. And I think that was the Pfizer drug. So the FDA is very interested in approving these drugs for this very prevalent disease. So what do these inhibitors do? So this over here, the long chain molecule here is a long hydrocarbon, is a fatty acid. That's the acid part. That's the hydrophobic part. Makes it makes a lipid. And it can get broken down, but it has to get into the mitochondria to get broken down. And it turns out that acetyl-CoA carboxylase is the enzyme that makes an inhibitor of the enzyme that gets these fatty acids into the mitochondria. So if acetyl-CoA carboxylase in green here is active, it will inhibit the oxidation, the breakdown of the fatty acids. And so what these drugs do is they inhibit the acetyl-CoA carboxylase. So, it's a double negative here. So if you inhibit the molecule that it's inhibiting, then you stimulate the breakdown of the fatty acids. And so what we've seen in our experiments is we see a decrease in the amount of fatty acids, and we see an increased amount of the product's ketone bodies. So, if you're starving for a long period of time, you start breaking down fat and you start to get ketone bodies accumulating in your blood. So the formation of ketone bodies is an indicator that you're breaking down fatty acids. So we've done many experiments with this now, but I just will show you, for a general audience like this, just show you a couple of examples. But one thing we did was we took mouse retinal pigment epithelium choroid tissue, basically the back of the eye with the retina removed, and we measured the production. We measured two things. We fed them fatty acid, and we saw it disappear, and then we followed the formation of ketone bodies from the fatty acids. And we wanted to know whether these ACC inhibitors, the Fursocostat and Clisacostat, were capable of working on this tissue. And it was actually very effective. This is a Fursocostat experiment showing that it's the breakdown of fatty acids and the production of ketone bodies. So these drugs are very effective on RPE tissue. And the other experiment I just wanted to show is one that we've done recently. Remember the structure of these lipoprotein complexes, that they have cholesterol in them, and the RPE cells. If you can grow human RPE cells in culture, and then you can measure how much of these lipoprotein complexes are released into the medium, and you can measure the amount of cholesterol in the medium, which is in the core of that. And you can also measure a protein. In this case, we're measuring a protein called ApoE. So the surface of this is covered with phospholipids, and it's also covered with proteins. And so there's ways to detect that protein. And so when we measure in the medium cells, but if we add this inhibitor, the cholesterol doesn't get released. And if we do the same thing, we look at ApoE, the protein that's bound to these lipoproteins, the ACC inhibitor, these are pretty low concentrations, like 100 nanomolar is a very low concentration. It's effective at very low concentrations at diminishing the amount of these lipoproteins that are being released. And one thing that was really interesting to us was that it's sort of surprising, was that if you look at the cells themselves, and so here, the blue is the nuclei of the RPE cells. So this is the monolayer of those cells on a dish. And the white is staining for that ApoE protein, the one that's part of this lipoprotein complex. And so the RPE cells make this ApoE protein, but normally you don't see it on the cells because it's being exported with the lipoprotein complexes. But if we stimulate the breakdown of fatty acids so that there's much less fatty acids in the cell, they can't make these lipoprotein complexes. They still make the ApoE, and they retain the ApoE in the cell. So we have a lot of evidence data like this that show that these drugs are effective at stimulating the breakdown of fatty acids, and that causes the cells to release less of the lipoprotein complexes. So, so how are we planning to use this? Our strategies for translating these findings to a treatment for AMD. So, like I said, these ACC inhibitors have been safe in clinical trials for fatty liver disease. It turns out that in none of those trials, they didn't cross their mind to really look to test to see if it affected people's vision. No one noticed that they had problems with vision, but they didn't actually do any specific tests in vision. So one of the first things that we want to do is we want to test these molecules in mice, and we're doing a bunch of biochemistry experiments like the ones I just described, but we also want to test their vision to see if it has any deleterious effects on the vision. Fatty acids play a very important role, many very important roles in vision. So we want to make sure that these drugs are not having adverse effects on vision. So we're doing that. Kathy Bose-Rickman at Duke University has a very nice animal model that accumulates Drusen material. So one of the next stages in this project is to work with her to test these drugs to see if they can diminish the Drusen deposits in an animal model. And then finally, like I said, the FDA has approved this for fast track. So it's not yet approved by the FDA, but our plan is that when these do get approved by the FDA for treating liver disease, hopefully we can then use them in clinical trials off-label to see if they can diminish Drusen and improve vision in people with AMD. And we have a couple of labs here that I think will be very helpful in making those evaluations. And finally, Chad asked me to write what would our wildest dreams be, and so if this project was successful, whether Drusen is, like I said at the beginning of the talk, is it just a benign symptom of AMD, or is it a cause of AMD, or is it some combination of both? So we'll gain more insights into the nature of the disease, and then if it works as well as we hope, we would have identified pharmaceutical agents that could minimize the Drusen deposits in patients with AMD. And if that is what's interfering with their vision, that might help them improve their vision. And then in cases where the Drusen has accumulated, make it go away. Maybe it could even restore vision in some cases of AMD. So those are our wildest dreams. But I'd like to thank my colleagues here at the University of Washington: Dan Haas, Jen Chow, Marcos Nazario, Abby Engel, and Rain Lim, and we're really looking forward to working with Kathy Bose-Rickman. And of course, I really want to express how grateful I am to the Foundation Fighting Blindness for supporting this project.
All right, Dr. Hurley, thank you so much for that presentation. We do have a few questions for you that we'll take now. I think one of the questions, I guess it's two parts to the same question, but number one, how would this therapy be delivered? And then since dry AMD is sort of, its disease etiology is through stages, at what stages do you think that this drug could be delivered? So how is it delivered, and then at what stages?
Yeah, so right now it's for the fatty liver disease, it's delivered orally. And so they're testing out many different doses, and like I said, it's so far been shown to be safe. And in the experiments that we're doing with animals to understand its mechanism, one thing we can do is we can with mice we can infuse very specific concentrations, and we can measure specifically the effects of the drugs on fatty acid oxidation in specific tissues. So, but that's to investigate the mechanism. But I think as a therapy, if all this orally. What was the second question?
Could you repeat that last part you just said? Our connection broke up a little bit.
Oh, yeah.
I believe you said it'll be administered orally. Was that the idea?
Yeah, that's the way it's being, that's the way it is being used in the clinical trials so far. It's been delivered orally for the fatty liver disease. And there haven't been major side effects. The biggest side effect has been for reasons I don't think they completely understand, but the triglyceride levels go up. But with the way the clinical trials are doing now are with this ACC inhibitor that I described. But they also found that if they co-deliver that with a drug that inhibits an enzyme that converts, that adds a fatty acid on to make triglycerides, if they use those two drugs together, they're very effective. And that's the combination drug that's being tested now. And most likely that's when we start testing these in the animals, we will also test that combination of drugs.
All right, our next question is, how long after sort of the initial treatment would you expect to see therapeutic effects or even visual restoration?
That's one of the reasons for doing these animal models. And I don't know what the answer is. I can imagine that so my understanding is that the Drusen is kind of something that is made, and sometimes it's there and sometimes it goes away. So it changes. And so if you can stop it from being made and it gets broken down, then the Drusen could go away. So I think if it is approved by the FDA and we are able to do clinical trials, what we would do is we would administer the drug probably orally and then monitor the Drusen deposits in the patients to see if they get smaller or if they move around. So I think it could be anywhere from within days to within 2 months. And we're just sort of hoping that that this drug goes through the FDA and we're able to do those clinical trials and get actual data on that.
Yeah, I think many people will probably be excited if we are successful in this, that it probably will be in an oral formulation. I think that's very important. The last question is, I think you mentioned that during the clinical trials of this particular compound in mitigating fatty liver disease, that no one really looked at its impact on vision. However, on the sort of flip side of that, do we know if people with fatty liver disease have an increased risk of age-related macular degeneration?
Yeah, I don't know the answer to that. And I guess, yeah, but one thing, just one comment though, one thing that's encouraging is that during those trials, no one had reported visual problems. So I'm guessing that if there are visual problems, they won't be very severe, but they might be something that could be detected by looking, at specific things like light or dark adaptation or things like that.
All right, we do have 2 more questions for you. However, we're going to hold these questions until the general Q&A session, just in interest of time. Dr. Hurley, thank you so much for the presentation. I think we learned a lot, and it's definitely, hopefully it's well received. Next, the presenter we have, our final TRAP awardee presentation is from Dr. William Beltran from the University of Pennsylvania. This is a unique award that we have awarded to Dr. Beltran and the team of researchers. This is one of our program project awards. And that means it's a large, multidisciplinary, multi-investigator project that addresses a critical knowledge or therapeutic gap in regard to inherited retinal degenerations and or dry age-related macular degeneration. This particular program project award came out from a directed research program that was designed through a topic-specific deep dive workshop, in this case from the virtual workshop on inflammation and viral gene therapy of the retina that was held back in 2020. This program is directed by a single PI, Dr. Beltran, with 2 other collaborators on the project. Dr. Beltran's research is focused on examining the cellular and molecular mechanisms of photoreceptor death in animal models of retinal degeneration and developing novel therapeutic approaches aimed at curing or slowing the progression of this group of diseases in human patients. Dr. Beltran is a board-certified veterinary ophthalmologist, and he consults at the Ryan Veterinary Hospital at the University of Pennsylvania and teaches clinical ophthalmology to students. Dr. Beltran, we are ready for your presentation.
Thank you very much, Chad, for the introduction. Can you hear me well?
Yes, we can hear
Okay,
you.
super, excellent. Well, thank you very much for this opportunity, and on behalf of my collaborators, Dr. Ragvi Sudas and Dr. Shannon Boy, I'd like to thank the FFB for funding this project. So today I'm going to let you know a little bit about what we're planning on doing, and the entire goal here is to try and understand a little bit better how and why a type of viral vector called an adeno-associated virus, or AAV, is capable of inducing ocular inflammation, which is probably not what we want, but it's what we observe when it is used to develop retinal gene therapies. And the entire idea is by better understanding this, we might find, in fact, approaches to try and mitigate this ocular inflammation. Now, before I get started and talk about inflammation per se, if this advances, which it is not, okay, there we go. I think this is working. I'd like to first kind of summarize what an AAV is and why it's being used in retinal gene therapy. An AAV is a virus. It's composed of a shell, if you want. It's called a capsid made of proteins, and within that shell is the DNA material that you want to maybe introduce into a cell. So it's a therapeutic DNA that you're going to introduce in a cell that might be missing a copy of a specific gene, or maybe basically producing a gene that is abnormal. I think what's really important is we're going to talk about inflammation, but the critical thing to hear about is that AAVs are the to-go type of viral vector currently in retinal gene therapy. You've all heard about the success of the Luxterna. You may not have heard that there are past and ongoing clinical trials, more than 30 of them, in patients. And there are many, many more, in fact, studies that are using animal models to try and develop gene therapies with this type of vector. So why is it being used? Because it's safe and because it expresses, in fact, low immunogenicity. Now, that low immunogenicity is relative, and I think that Chad mentioned that there was this workshop organized in 2020, and subsequently a paper that was published, a white paper. And what is very clear is, by bringing a group of experts in a room, that yes, injecting an AAV can cause some inflammation, sometimes severe, sometimes mild, and that there are multiple causes for that to occur. So the following slide is kind of listing the type of culprits that have been identified and potential strategies to mitigate that AAV-induced ocular inflammation. First of all, the AAV by itself, whether it's the proteins on the capsid, whether it's the DNA material within the cell, can cause inflammation. And so basically finding new capsids or finding sequences that are less pro-inflammatory is what some groups are trying to develop. The dose is always what makes the poison, regardless of the drug. So if you're able to identify AAVs that are more efficient, or promoters, which are kind of switches that turn on the DNA, the therapeutic DNA, in a more efficient way, you might be able to lower the dose and therefore reduce the inflammation. There's a lot of effort that has to be made by those who make these vectors, and then from an industrial standpoint, in basically producing products that are more pure, that have less contaminants, less residuals, that have less viral vectors, that have only a shell and no DNA inside. And finally, there are surgical considerations. The site of intraocular delivery is critical, and it's known that depending on where you deliver those viral vectors in the eye, you may have a more or less important inflammation. If you injected these viruses in the vitreal cavity, which is the gel that is inside the eye, the vitreous, it tends to elicit a higher immune reaction than if you inject it in the subretinal space. And since for most of these diseases, injecting the subretinal space is the place to actually go if you want to target photoreceptor cells or the retinal pigment epithelium, there might be ways of retaining that virus in the subretinal space and avoiding any leakage elsewhere. The surgeon can potentially be responsible. And depending on the ability, technique, experience, there is definitely some reasons here for potentially having induced inflammation. And so some groups are looking at ways of replacing the surgeon by a robot, or using injectors that have a controlled release system. And finally, last but not least, the patient, the age, stage of disease, the immune status, and all that to say that we're missing a lot of information or knowledge on the disease and the immune status of individual patients. And the reason being because we're lacking biomarkers that are easily accessible, that can allow us to know what is the status of that patient before we treat and after treatment. So our goal, and I'm going to summarize, not to tackle all these challenges. I think that's something that multiple groups are doing, but I've highlighted here the three, and I'm going to expand on each of the next slides on those three goals. So the first goal, and that's a project that is led by Raghavi Sudarsan. She's a research assistant professor at Penn within our group, and she's had for quite a while an interest in retinal inflammation. And what basically she wants to do is try and identify specifically these biomarkers that are easily accessible and that could actually allow to monitor that inflammation caused by AAVs. Now, there are problems. The problems, as I mentioned, one is that we know AAV vectors can induce retinal inflammation. That is known. But the other problem is that retinal degeneration on its own can cause inflammation. And several groups have looked at that, and Dr. Sudarsan has done so in canine models and shown, and what this figure is showing you is nothing else than bars and colors, but it's showing you the genes that are upregulated, meaning expressed in early stages and late stages of retinal degeneration in canine models. And that's been shown in multiple animal models. It's shown, in fact, in people, that there is, in fact, an induction, in particularly of the innate immune system with disease itself. The other problem, which I mentioned, is that there is really no good non-invasive biomarker to measure that ocular inflammation. So what is the approach? She wants to try and characterize a little bit better the immune response caused by the disease itself, but doing so at the single cell level using a technique that's called RNA transcriptomics, or single cell RNA sequencing. And the entire goal here is, instead of looking at the big picture of what's happening in the retina from an inflammatory standpoint, is looking at individual cell populations. What's happening to rods? What's happening to cones? What's happening to microglial cells, which are resident immune cells? So in a certain way, it's like going to a concert hall, and instead of listening to all the instruments together playing the symphony, it's picking up specific instruments, the strings, the percussions, the brass, etc., and identifying whether these specific instruments are in tune, are playing a sad melody, are stressed or not. And by doing so, she aims basically at understanding better what is going on in disease at the single cell level. That's the first goal. Second goal is, once that's done, is to now look at what happens when you deliver an AAV to the retina. What happens, and you deliver to a normal retina or to a retina that's diseased? What are those inflammatory pathways, cascades that are being activated? So just to summarize, here, the main goal is being able to dissociate what is disease-related inflammation from what is AAV-induced inflammation. Why is that important? Well, because once you've identified potentially some markers, some pathways of inflammation that you know are viral vector specific, you may be able to identify what those genes are, what those genes produce, and they produce a protein. And we may be lucky in identifying proteins that are secreted. Secreted where? Elsewhere than in the retina. They're going to be secreted in places in the eye that are easily accessible. Unfortunately, we cannot do a biopsy of the retina. That's something that is not possible. But we could do a fine needle aspiration of aqueous humor, or maybe a little bit of vitreous, and identify some of these proteins that are pro-inflammatory, that are upregulated. And that would provide a tool, basically a biomarker, to assess the status before gene therapy as well as after gene therapy, and if there is inflammation in a patient, hopefully try and modulate that as we go, rather than waiting for having severe clinical signs following the gene therapy. So that's goal number one, or module number one. The second module, which I'll be leading, is to try and evaluate whether there's a better way of delivering the viral vectors to the subretinal space and hopefully decreasing the possibility of having any induced ocular inflammation. The current problem is that the main route of delivery for the vast majority of the clinical trials and preclinical work that's done in animal models is through what's called the Trans-Vitriol subretinal injection. That's the kind of standard to deliver an AAV to the subretinal space. So what I'm showing you is here is an image diagram that shows the surgical procedure. And what you do through this approach is that you introduce a needle inside the eye in the vitreal cavity. You approach the retina, you make a tiny little hole in the neuroretina, it's called a retinotomy, and you inject the viral vector solution. So you're detaching focally the retina, delivering the viral vector, and this, and obviously you retract the needle, and within roughly 20 to 48 hours, this localized retinal detachment is going to reattach. So you've got an area, localized area of treatment. Now that's what's done, that's what most groups do. The problem, and we all know about it, is that there is some degree of leakage through that tiny little hole of the viral vector solution that you've injected. So you're losing obviously some of the solution, but most importantly, you're making now that AAV more easily accessible to the immune system, because in the vitreal cavity, the immune system is better able to recognize these viral vectors than in the subretinal space and to mount, in fact, a immune reaction. And we know this from various studies. So what is the approach? The approach is basically to try and access the subretinal space, but from behind, instead of coming from the front. Using this device that has been called the Orbit Subretinal Delivery System. It's been developed by a company called Gyroscope, now in the hands of Novartis. And the surgical technique is slightly different. We basically make a small slit on the sclera, which is the white shell of the eye, and introduce this white cannula, as you can see, in what's called the suprachoroidal space. This is a space between the sclera and the choroid. So we're under the retina, if you want. And once we're at the right location that we want to treat, there's a little knob. And what we do is that we advance a needle that's at the tip of that cannula that's going to enter the subretinal space from behind and allow us to deliver the viral vector solution. And then obviously the whole system, the whole cannula is retracted. So the purpose of this module is going to evaluate whether we can use this device in canine models at different ages and stages of disease. And if that happens to be the case, then what we're going to do is compare this approach to the Trans-Vitriol standard approach and see whether by using this approach and avoiding this reflux into the vitreal cavity, we're going to be able to reduce the probability of having, in fact, any ocular inflammation. Finally, the last module that is led by Shannon Boy at the University of Florida is aimed at developing and testing new AAVs that have the ability to disperse better, to spread, if you want, tangentially. And the whole idea here is potentially being able to treat a larger area with a potentially lower dose. So what is the current problem and what is currently done? The current problem is, as I showed you on the previous slide, when you detach the retina and cause a subretinal bleb, basically that's the area that you've treated and that's it. So it's the treated area is really dependent on the size and the location of that subretinal injection bleb. And what you see here is a photograph of several photographs of a non-human primate, so a monkey, that was injected with a classic standard AAV, and the yellow line shows the border of the bleb, and you see over time an increase in fluorescence that appears white. This is a fluorescent protein, but you see that fluorescence is limited to where you did the bleb. It doesn't expand. Now, if you want to treat, for example, this region right in the center, which is where the fovea is, and there are diseases that affect the fovea, you've got to create a bleb right in the fovea, and the risk is you can potentially have visual acuity loss or retinal thinning when you detach the cone-rich fovea, which is a very delicate region. So what is the approach? Well, basically, it's to identify and to test new AAV capsid variants that have this increased ability to disperse, that can hopefully allow us to use a lower dose and can allow us to target the fovea without, in fact, detaching the fovea. So what we're going to do is, through the course of those 3 years, is to test 3 such lateral spreading AVs that have been developed in the Shannon lab, and we're going to compare them to a classic standard AV, and I hopefully identify a winner, what we call the lead candidate, and then subsequently compare that winner to the classic AV in a model, a well-characterized canine model of inherited retinal degeneration, and see whether basically we can do the same thing, decrease the dose of virus, and hopefully decrease the inflammation that we see with some of the standard viral vectors. So just to summarize, this is a figure, a little complex figure, but it basically lists the various modules. I think what is important here to see is what are the deliverables. So what we're hoping to achieve is, first of all, identifying retinal inflammation biomarkers that are easily accessible. 2 is to hopefully identify a route of injection that lowers, in fact, the ability or the possibility of seeing any inflammation. And finally, using some AV vectors that have an ability to spread more, to therefore treat a larger area and hopefully decrease the dose. And you can see that these are 3 modules that are going to be highly interactive. And with this, I'd like to thank you for your attention. And again, thank the Foundation Fighting Blindness for this support for this exciting project. Thank you.
Dr. Beltran, thank you so much for that wonderful presentation. And up front, I want to wish you and your team good luck in doing this because it's so important. I think I'll take the first question for you because I want to highlight to our audience that this is the reason that this project is so important is because it spans sort of retinal therapies, not only maybe not even for the retina, but also will give evidence to maybe other fields as well as how to mitigate the inflammation caused by AAV-based therapies. I guess, is there a particular point of the project that you're most concerned about that we have to get over or have to get past in order to be successful?
I think these various models have their own challenges. And we have evidence that, for example, with the subretinal injector, we are able to deliver viral vectors in wild-type retinas, so normal retinas. The question is, are we able to detach now a retina that is at, for example, a more advanced stage of disease? How easily is that? And I think that's one potential challenge. The challenge for the retinal inflammation biomarkers, it's really we don't know what we're going to be able to fish, and are they going to be basically some potential proteins in levels high enough in for some of those fluids, ideally the aqueous humor. That would be the best one that we can measure using, and these are technique of mass spectrometry, etc. So that's a potential challenge. Then regarding the AV capsid, the spreaders, we know for sure, and this is work done by the Boy Lab, that yes, these viral vectors do spread. The question is, by spreading more, can we now lower the dose? And if we really do a side-by-side comparison with a classic AAV, are we able to treat a larger area with less virus? And that's currently unknown. So I think there are definitely challenges, and I think also what I mentioned on, I think, my third slide were the list of other culprits and potential approaches. So I think that this is, by no means are we going to be addressing every single of these problems, but at least I think if we're able to address a few of these, that will already be a major success.
Okay. The next set of questions are: how often do you get a retinal detachments during or after retinotomy, or is there evidence of choroidal hemorrhage using the scleral approach?
Yeah, so retinal detachments, in our experience in animal models, extremely rare. Even when we are intervening in stages of disease that are very advanced, where the retina is very thin, where there are few photoreceptor cells, we do see, in fact, a reattachment. So that's good news. Now, in patients, it's slightly can be potentially different. It's not so much retinal detachments, it's more holes in the foveal region that have been observed in some clinical trials, also in fact in preclinical studies in monkeys. And having a hole in the fovea, which is the region that is the really critical for visual acuity, is obviously something that you're trying to absolutely avoid, and thus the reason for trying to test some of these AAV capsid variants that are going to allow us to create a bleb away from the fovea, but still have the virus over the course of several weeks migrate and reach, in fact, that foveal region.
All right, thank you. Next question is, what is the size limitation on viral constructs crossing Bruch's membrane when administered via the suprachoroidal delivery?
Yeah, it's the same as for the classic approach, and the reason being the viral construct, there is a limitation indeed to the amount of DNA that you can put in an AAV. It's about 4.7 kb, whatever that the measurement is. That's the unit of measure if you want. But whether you introduce the viral vector into the vitreous in the subretinal space, or via the suprachoroidal approach to then deliver it into the subretinal space, at that point the construct is within the AAV. It gets opened up and freed once it's in the cell. So really, there is no more limitation depending on regardless of the route of delivery. Basically, the limitation remains the same, which is that of an AAV. And there are obviously some diseases that for which you would have to introduce a much larger gene, and that's why some people or some groups are looking at other type of viral vectors that have a larger, what we call, cargo capacity.
All right, thank you. When you mentioned about the sort of the AAV type and basically developing something that can spread laterally throughout the retina, I think that's a very exciting proposition for the field. I think maybe the one question that has come in that maybe gets at this: does the retinal disease severity, could that impact a new AAV's ability to spread across the retina?
Yeah, so that is important point. What these AAV capsids, so variants, have been modified to be less sticky. That's basically what it is. There are in the retina, in the subretinal space, there's what's called an extracellular matrix that is in fact a bit of a barrier for those AAVs to migrate. And these various capsid variants have been modified and been mutated to not stick as much, and that's what provides them the ability to disperse. I think the point is very interesting. It is a good question, because we need to understand, and we have little understanding of what is that extracellular matrix in advanced disease. And I think the work that we're doing, that we're proposing to you to obviously test these in animal models that are at various stages of disease, is precisely to see whether or not changes in the composition of that extracellular matrix may or not prevent, in fact, that spreading ability for these viruses. So that's an excellent question.
All right, we have maybe one more question for you before we open it up to the rest of the presenters. We have a question here that: how does this therapeutic approach potentially impact a person with Stargardt's disease?
So this therapeutic approach for Stargardt's disease is, or for anything, is, I would say, in the case of Stargardt's is an example of a disease caused by mutations in a very large gene. And that's why some groups have been trying to develop treatment using another type of virus, a lentivirus, and success has not been there. So if groups are trying to develop AAV-based gene therapies for Stargardt disease by either trying to find a smaller version of the gene that you can introduce, or cutting the gene in half, putting one half in a virus, putting the other half in a virus, co-injecting both, and making sure that there's basically a tape, if you want, on the pieces of DNA, so that when the two viruses infect the photoreceptor cell, they can reconstitute a normal length gene, ABCA4 gene. So I think that all of these approaches, whether it's measuring the inflammation, whether it's better delivering, or potentially developing better AAVs, would be applicable to Stargardt, with the caveat that we're dealing with a larger gene.
All right, thank you. And I want to throw one quick question. It looks like, man, we're having so much fun here. I think we're coming up on our time, but I would like to throw a question to Dr. Adder to maybe weigh in. So we talked a lot about inflammation, and during your presentation, we talked about basically taking cells and putting them into the retina. Do you think inflammation is going to be a big concern for this particular therapeutic approach? And how do you see maybe us getting around that in the clinic?
Yeah, absolutely. I think that's very important. And as was said, already said, there is some inflammation also after transplantation of cells. And that's why I mean, one idea is if you use induced pluripotent stem cells, some labs go, individually. So you can take or make an induced pluripotent stem cell from a patient and make their own photoreceptors or RPE cells. But we think that is, due to high costs and long development times and everything, that approach is very difficult to really bring it to many people. So that's why our idea is to use the super donors. So that means you make a bank of iPS cells that can cover a big number or percentage of the population. So you don't need for every individual one, but you have the super donors that can be used for many different patients. And that's exactly the approach we want to test and want to see if we can generate from these HLA homozygous lines in this direction. And then we hope that we can use these without inflammation or immune rejection in patients, in many different patients from one line.
All right, thank you. And I would like to thank all of our TRAP awardees. Congratulations. Looking forward to engaging with you throughout the year and throughout your different projects to learn more and help advance therapies towards the clinic. Now we will have closing remarks from the Foundation's Chief Scientific Officer, Dr. Claire Gelfman.
Thank you very much, Chad. And I really want to thank all of the presenters today. We reviewed a lot of different applications for TRAP awards, and it's no surprise through the work that you described today how and why it's so important here at the Foundation. I was just reflecting in our 50-year history as you all were talking, and then those very early days, the awards that we funded were about identifying new genes, the genetic components. Well, you'd go to your doctor, you get a diagnosis of, let's say, retinitis pigmentosa or AMD, but you really didn't know what the genetic cause was. And so it's no surprise then, during those early days, we were all about funding the identification of genes when mutated resulted in that clinical diagnosis you get from your doctor. And so we at the Foundation, though, have evolved. We have funded work in over 100 genes now of about the 280 that we know that are sort of classified as those to be inherited retinal disease genes. And happy to report, as you all know, we've heard a little bit today about Luxterna, which is the only gene therapy for the eye that's been approved by the FDA. And that's incredible. And we're funding more work in sort of the gene-specific, gene replacement, if you will, world. And in fact, that field has evolved. And we heard beautifully from Dr. Beltran a moment ago about how we are looking at the safety now, the advanced safety of using those viral vectors, those adeno-associated viral vectors that he so beautifully described. Because as more work is done, we want to improve upon that safety, and we want to improve upon some of the inflammation that we know that gets induced sometimes during that therapy, and specifically figuring out, is it due to the disease itself, or whether due to the product, the medicine, if you will, the AAV gene therapy. And so we look forward to the success of this program through the funding of the TRAP. Now, in addition to sort of the gene-specific therapies that we talk a lot about at the Foundation, we want to increase our shots on goal. We want to increase the chances of getting things through the clinic to an approval for both inherited retinal diseases as well as the dry form of age-related macular degeneration. And not all of us have the privilege of knowing what gene is mutated that's causing our vision loss. And so what excited me today as well is about 3 other approaches to treating photoreceptor degeneration in a way that's not necessarily dependent upon knowing your causative gene. And so we learned a lot about some transplantation biology. So Dr. Adder showed really nicely how stem cells are being used to derive a population of both retina and these retinal pigmented epithelial cells that normally reside in the back of the eye. And when they degenerate, they die, and sometimes they're gone. And it can obviously, compromise vision tremendously. So no matter what the genetic cause, it's photoreceptor degeneration for your rods. If your rods and cones are not able to trap light coming in and send a message to your brain that interprets what that image looks like, then you can't see. So by restoring that environment, by transplanting this population of cells that are a combination of both photoreceptors and those pigmented epithelial cells, that transplantation will be a great way to think about using stem cells as a way to make cells that can actually help the cells in the back of the eye regain their function. We also heard today from Dr. Chen, who showed an alternative to cell transplantation, where instead of using an approach that could be, that may have some surgical implications, an alternative to cell transplantation could be to reprogram the cells that are already there. And so we learned about how epigenetics is being used to reprogram the endogenous cells, the cells that are already in the retina, to induce regeneration. And that as an alternative approach to actually cell transplantation, both of which have the potential to treat a degenerating photoreceptor that leads to blindness or compromised vision. So I mentioned before that another scope that we fund here at the foundation is for the dry form of age-related macular degeneration. And you can actually have dry AMD and not even know it. And you can go to your physician, your ophthalmologist, just for sort of your yearly updated appointment. And your physician may notice these yellow spots on the back of your eye, and that's called Drusen. And if you build up too much Drusen, it compromises vision. So no surprise then that a therapeutic approach that many, including Dr. Hurley, is taking is to reduce the amount of Drusen, to get rid of it or prevent it from forming to begin with. And so in order to do that, we need to learn a lot about that Drusen. We need to figure out what it's made of, how it's formed, so we can then think of a therapy to reduce its formation or perhaps inhibit it once it's built up. And so we heard from Dr. Hurley some work that he's done on the role that fatty acids play in the development of Drusen, and we heard also about an inhibitor that, by the way, is already in human clinical trials for an alternative non-ocular indication. And based on the mechanism, it's got the potential to be used to inhibit that Drusen formation and thereby inhibit the progression of the dry form of age-related macular degeneration. And interestingly, that particular therapeutic is designed to be given orally, so by mouth. So, most of the treatments we hear about are given directly into the eye, whether it be subretinally or intravitreally, so under the retina or right inside the middle of the eye. But again, to sort of expand on that toolbox approach, we want to think about different ways to treat vision loss. We also want to think about different ways to deliver therapeutics once discovered. So what you heard about today are really beautiful examples of the funding that has come through our TRAP program, this Translational Research Acceleration Program. And it's a mechanism of funding that's a little different from some of the earlier phase work that we fund. This is work, as you can see, that's got a lot more legs, if you will. It's even closer to the clinic. It's even closer to that first in human study. And so we look really forward to hearing the progress from all of the research which from all of the researchers that we heard about today in an effort to move these therapeutic approaches further down that will eventually lead to a potential treatment for different types of inherited retinal diseases, as well as the dry form of age-related macular degeneration. So I want to thank everyone for joining us today. It's been a pleasure having you with us as we reviewed our TRAP awardees. Many of you put in questions that we were not able to get to today, so please email them to us at info@fightingblindness.org. That's info@fightingblindness.org. Thank you again and have a wonderful day.